and we going to continue talking about the membrane potential and the ionic effects on the cellular membrane that happen when the signal is transmitted through the neuron and between the neurons. Okay, so I want to remind you a few things about what we were talking last class. We had a conversation about graded potential. Okay, when some kind of event on the membrane, some kind of a stimulus, opens sodium channels, influx of sodium into the cell depolarizes the membrane, which means membrane potential changes, and that change in the membrane potential opens voltage-gated sodium channels adjacent to the site of depolarization, adjacent to the site of the stimulus, and graded potential starts to sort of self-propagate along the membrane. However, since every process like this, every process of opening channel is accompanied by some energy loss, eventually graded potential will go away, will dissipate. That makes sense so far. Now, a few important things, a couple of important things about graded potential. First, if graded potential goes over the certain threshold, it becomes so-called action potential. So it does not dissipate anymore. When the level of depolarization, of the membrane depolarization, increases above the certain level, we have the action potential. We have a self-propagating signal that travels down the axon. And another important thing about graded potentials, they can sum up. They can add to each other. We're going to talk about it as well. So with this in mind, we will start talking about action potential. And as I mentioned, action potential is formed when the depolarization of the membrane exceeds a certain threshold level. In this example right here, you can see threshold level at negative 55 millivolts. This number is close to the real number, but it will be slightly different for different cells, depending on the ion concentrations in the cell, outside of the cell. This number may vary a little. Right? Are we clear on that? So let's, first of all, let's take a look at both what's going on in terms of the ionic flow when the membrane potential is formed and propagated, and how it is reflected on the graph. Let's take a look at this graph in the upper right corner. On the x-axis, you can see the time, and you have to appreciate this. This graph represents what is going on at a very small, very tiny patch of the membrane. Does that make sense? So we're looking at the membrane in one particular place. We are not looking at the entire axon, we're looking at the small patch of the membrane. Are we agreed on that? Awesome. On y-axis, it's the membrane potential, okay, in the millivolts. And what we start with, we observe the membrane. What we start with is the resting membrane potential, negative 70. And then voltage-gated ion channels, or it can be a stimulus. Well, but usually it's voltage-gated ion channels, and you will see white voltage-gated ion channels. Voltage-gated sodium channels, actually. They open, okay, due to that initial change, and you can see that sodium starts to flow into the cell. Okay, that's the part of the graph number 2. In the picture to the left, you can see that sodium channels are open, while potassium channels on the membrane are closed. The concentration of sodium is higher outside of the cell or inside? Outside, yes. So sodium diffuses, passively diffuses into the cell. Sodium is how charged? Positively. So positively charged sodium ions get into the cell and they increase the membrane potential, making it less negative or more positive, whatever characteristic you want to give it. And if it's going to be the action potential, then once the depolarization crosses the threshold level, this change in the membrane potential will become an action potential. So membrane potential changes, membrane potential elevates due to the influx of sodium all the way up to approximately 30 millivolts. Now, I do not require the memorization of those numbers. Don't worry about this. Really, I'm not going to ask you what is the voltage at the peak of membrane potential. Okay? However, what I want you to understand that at the level of 30 millivolts, at the peak of membrane potential, sodium channels close. At the same time, potassium channels open. Concentrations of potassium ion are higher inside of the cell. So potassium starts to passively diffuse out of the cell. Does that make sense? It keeps on diffusing, and potassium is charged how? Positive. So when positive ion leaves the cell, what happens to the membrane potential? It becomes more negative. And this process, this leg of a graph, is called repolarization. Does that make sense? Eventually, membrane potential will reach the resting membrane potential level. Okay, sorry, showed you the threshold I need to show. So resting membrane potential level right here. Okay, that's RMP. Well, hypothetically speaking, at this moment, everything should become normal, right? I mean, cells should go back to RMP levels. However, the problem is that potassium channels, well, it's not a problem, that's life, okay? Potassium channels are slow. So even when the cell reaches resting membrane potential, potassium channels fail to close yet and keep letting potassium out. So positively charged ions keep leaving the cell, and that creates this little overshoot. The part of the graph number 4, can you see that? That's the moment when the membrane potential of the cell is lower than resting. Does that make sense? This is called hyperpolarization. You see, potassium channels are still open, and sodium channels are not. You can see that little gate here, closed. Does that make sense? Yes. Mm-hmm. Yes. Close. Open. Close. They become closed when the cell gets all the way, so they are supposed to be closed at this level. But since they are slow, they do not close yet. They kind of wait a little bit. Does that make sense? So potassium keeps leaving until the potassium channels finally close at the hyperpolarized state. So it just lags a little bit. Okay? And in this situation, right here, during hyperpolarization, potassium channels will be closed at the very bottom of the graph, and sodium channels will be closed at the bottom of the graph. They both closed. And sodium-potassium pump will reestablish the resting membrane potential. Does that make sense? We have it here. So sodium-potassium pump, this structure will reestablish the resting membrane potential. It will pump the ions back to normal levels. Does that make sense? Excellent. Okay. Now, what I want you to, we didn't finish with this yet, but I will ask you questions. Something like, what happens to ions, certain ions during depolarization, repolarization, hyperpolarization? When sodium flows into the cell, how does the membrane potential change? How do we call this? Is that repolarization or depolarization or hyperpolarization? Which channels are closed at each stage? So you have to understand that ionic flow pretty well. And that is not an incredibly hard task, because you have two ions and you have three parts of the graph. All you have to know is which way they are going and what's going on with their channels. In fact, this graph will help you to better understand what happens during the depolarization, repolarization, and hyperpolarization of the membrane. So if you look at the purple line right here, purple graph, it represents the change in the membrane potential of the cell. Can you see that? Can you see that? The purple graph. You can see the resting state. You can see the depolarization. You can see the repolarization, and you can see hyperpolarization. Now the tiny little lines, the red one and the blue-green one, represent the flow of ions across the membrane, permeability of the membrane to a certain ion. For example, during depolarization, number 2, you can see that sodium permeability goes up. It is shown here. During repolarization, sodium permeability goes down completely, but instead potassium permeability increases. And you can see that potassium permeability stays elevated even when the cell reaches the point of resting membrane potential. That is why right here, due to that elevated potassium permeability, potassium ion keeps leaving the cell, leading to this overshoot, leading to hyperpolarization. And eventually, when the potassium channels and sodium channels are all closed, sodium-potassium pump re-establishes the membrane potential to the normal resting levels. Does that make sense? Go ahead.
So, I guess in some ways is that contributes to why all the cells are negatively charged?
All the cells are negatively charged, so resting membrane potential is negative. Because cell is much more permeable to potassium than to sodium. So the Nernst potential for potassium, as we figured out, negative 94, Nernst potential for sodium is plus 61. If contribution of both ions to the resting membrane potential would be equal, then it would be negative 33, negative 94 plus 61, okay? But the contribution from sodium is much smaller. Does that make sense? So the resting membrane potential is lower than negative 33. It's at negative 70 because potassium, flow of potassium out of the cell plays much more important role than flow of sodium into the cell. One more thing, you have to understand that which channels, what type of channels contribute to the resting membrane potential based on gating. What types of resting membrane potential? Are they gated at all? It's leaky channels. It's leaky channels. It's a potassium leakage out of the cell and sodium leakage into the cell are two ionic flows that contribute to the resting membrane potential the most. That's one thing. Action potential happens because of voltage-gated channels, which is a totally different thing. Does that make sense? So imagine pretty much this whole shtick right here happens because of the voltage-gated channels. Once the cell gets back to the resting membrane potential here, leaky channels will, they keep leaking. Resting membrane potential is being maintained. It keeps going. They're still leaking. Those are two different entities. Does that make sense? Okay? Think about this. Okay. I'm a sucker for analogies, and some of my analogies suck, pun intended. But imagine the store, like Walmart. You look at Walmart like any time of day, anytime, pretty much any time of the year, people go in, people go out. Okay? There's certain flow of people in, certain flow out, nothing unusual. But imagine they announce they have like 70% discount on TVs, for instance. So the moment, or it's a Black Friday, there's a rush of people getting into the store. Okay? So something triggered that move of people. And then everything is gone, everything is bought, people start to flow out. Does that make sense? It's the event that's separate from normal flow of people into and in and out of the store. So action potential is the sort of separate flow of ions mediated by different channels. Does that make sense? Awesome. Now this picture just kind of shows you what we are looking at. First of all, this is the electrode inserted in the axon. Let me tell you something. You can do that. You can take an electrode, like a little, small, miniature, tiny glass thing, and stick it into an individual cell, under the microscope, of course. And then you can measure the change of the membrane potential using the voltmeter. Pretty simple device, okay? So when the signal runs through the axon, before signal arrives, the membrane potential that is measured by this electrode is resting. Once that wave of depolarization comes through, you can see the membrane potential becoming less negative. That's depolarization, okay? And then, wave of depolarization passed. Sodium channels are closed, potassium channels are open. You see re and hyperpolarization. at the place where electrode is. So you have to understand what we see as the wave is the observation of potential changes in one particular place in the membrane. What actually happens is the wave of depolarization that travels down the axon. Does that make sense? And only when it's action potential, then it's sustained. Graded potential, it's not a sustained thing. It will dissipate. It will just disappear eventually. In case of action potential, when it is going, it's going to reach very end of the axon, and it's going to lead to the release of neurotransmitter, and it's going to stimulate, or not, the axon, the neuron downstream. And if it stimulates the neuron downstream and generates action potential on that neuron, then the signal is going through. Does that make sense? You see what I'm trying to say? Now, action potential has a critical, a very important feature. Its level, its value is the same all the time. Okay? Same electrical level. Action potential operates on the principle all or none. Does that make sense? I sometimes use the boxing analogy. Think about the knockout in a boxing match. If the fighter doesn't get up until the count of 10, it's a knockout. Does that make sense? And it doesn't matter if the punch was so hard that the guy ended up in a hospital, or the guy managed to stand up on count 15. The match is still lost. Okay? So action potential is always the same level, no matter what is the stimulus strength. Do you understand? Which question should you ask? If its action potential operates this all or none principle, it's always the same level, no matter what the strength of the stimulus is. Nothing sounds wrong here? Okay, so pretty much the question, how can we distinguish between the strong stimulus and weak? And we definitely can, right? You can distinguish if the pain is tolerable or not, right? You can distinguish if it itches like hell or you can tolerate it. So we can do the discrimination in terms of the stimulus strength. Or think about this. When you contract a muscle, is that a nervous stimulus that reaches the muscle? Sure, yeah, if it's skeletal, right? Can you make a contraction stronger or weaker? Yeah. If I'm lifting this, I don't need much strength, okay? If I'm trying to lift this, I won't be able to, but I need a lot if I want to try. Does that make sense? You see the difference? Even if I lift this, I need to produce much more effort compared to this. So I can change the response, I can change the stimulus to the muscles. But level of action potential doesn't change. So what happens is, based on the level of the stimulus, frequency of action potential is going to change. Does that make sense? And, go ahead. Yes, and let me explain it using the muscle. So think about this. You have a bunch of fibers. We talked about structure of skeletal muscle, and that should be enough for now. You have a lot of muscle fibers. Does that make sense? In your muscle. And each of these fibers can contract. And when I need to lift my phone, I need only so many muscle fibers that have to contract at pretty much very infrequently, or Some may contract now, some may contract a little bit later. Okay, so frequency of contraction isn't going to be high. But if I need to lift my laptop, I need more fibers to contract more frequently to produce that effort. Does that make sense? So the only thing that changes is the frequency of the signal that goes to my muscles through my nerves. You see what I'm trying to say? So you have to think about it. It's always about the frequency, the change of frequency. I can tell you even more. When you hear sound, sound can have different loudness, right? The only thing that changes in your brain is the frequency of action potentials that go from your inner ear to the auditory cortex. It's not the level of the signal, it's how frequent that signal gets into your brain. Does that make sense? It also explains actually why some sensations just don't have them. Think about this. If the stimulus, whatever it is, if it's maybe it's a touch, maybe it's a sound, maybe it's a light that gets into your eye. If the stimulus is weak and the depolarization of the membrane that it causes doesn't reach the threshold, there's going to be no membrane potential. Does that make sense? If there is no membrane potential, are you going to feel the stimulus? Are you going to detect it? You won't be able to, correct? Only when the stimulus is strong enough to cause the depolarization that crosses the threshold and generates action potential. Do we understand the idea of all or none and increased frequency? Okay. Now, when the action potential occurs, now think about this. You never asked me one question. You look at the graded potential, and this picture clearly shows that graded potential can spread in both directions from the side of the stimulus. Can you see that? Yeah, like a ripple. Yeah. You look at this, an action potential most definitely goes only one way, through the axon, right here. Goes this way. The question should be, what the hell's going on? Why it's not going the other way? The answer to this is the presence of two refractory periods. Before we talk about them at all, I'm asking you, do you understand the concept of refractory? What is refractory? When something is refractory, can you give me like a very simple explanation? When I'm refractory to an infection, what does that mean? I'm not sensitive for some reason, okay? So being refractory means that it's not sensitive. It does not respond. There's no change. Does that make sense? So one refractory period that pretty much matches the depolarization and the start of repolarization is called absolute refractory period. And to understand what's going on there, we have to go back a couple of slides and look very, very closely at the sodium channel here. You may notice that sodium channel has two gates. One gate is represented here as the little ball, and another gate is represented here as, I don't know, like two thingies in the channel. So this ball is the true voltage gate, when membrane becomes slightly depolarized, it opens. Does that make sense? When it becomes hyperpolarized, it closes. More positive opens, more negative closes. Works pretty well, huh? Now, at the height of the... No, the wrong way. The height of the depolarization, at the peak here, sodium channel isn't just closed. It is inactivated. So if we go back, sorry about that. Here we go back. Can you see this gate being closed here? So that inactivation means that nothing, really, just nothing, can open the channel. It just stays closed. Does that make sense? I usually compare it to opening the gate at the parking lot. Imagine that you work somewhere and there's a parking lot with a gate, okay, and you come in and you swipe the card, the gate opens, you walk in and you drive in and it closes. Usually what happens, if you want to open it right away, you cannot. Your card is sort of inhibited. Does that make sense? So you cannot just park like 15 cars after you entered. Does that make sense? So that is the moment when sodium channel, during absolute refractory period, sodium channel isn't just closed. It is totally inhibited. Nothing can open it. Nothing. Am I clear? Nothing. So that makes sure that action potential cannot go backwards. Sorry. Action potential cannot go backwards because sodium channel here cannot be open, so that wave of depolarization can only move forward. Cannot open the channels behind it because they are inhibited. They are totally closed. Nothing can open. Not a single kind of depolarization can do that. Does that make sense? That's absolute refractory period. So it's just nothing, no stimulus, no exception. Does that make sense? Another sort of safeguard protective mechanism that ensures that depolarization, that membrane potential wave, the action potential wave, moves in only a single direction is the relative refractory period. So it occurs here during hyperpolarization. So here's what happens. Potassium channels are open and potassium flows out of the cell. Does that make sense? When potassium flows out of the cell, what happens to the membrane potential of the cell? It becomes more and more negative. So in order to depolarize the cell, you now need to apply the stimulus much higher than the normal one. You see what I'm saying? So if, say, this is the threshold, if you need to depolarize the cell from the level of resting membrane potential, that's the depolarization you have to produce. If you need to depolarize the cell from the level of hyperpolarization, that's the level you have to produce. Does that make sense? It's like, like when you're lifting something from the floor, it's one effort. If you try to lift the same weight, but from the hole in the ground, it's totally different effort. You have to produce, bigger, you have to make, you have to do bigger work, okay, more significant work. Does that make sense? Now, in case of relative refractory period, if the stimulus is stronger, okay, it can produce action potential. Does that make sense? If you want an example, I can give you one. Think about this. You send the signal to your muscle. in attempt to lift this laptop. And obviously the signal is not enough to lift it. You can see I'm not lifting it. So I send consciously stronger signal that overlaps with the relative refractory period on the neuron. And since it's stronger, it produces action potential and produces this elevation. I just want to show you that the effort increases, okay? So I kind of start to produce action potentials at the increasing frequency. They're all supposed to be of the same level. Sorry, my artistic capabilities are absolutely horrible, okay? They're all supposed to be of the same level. So if I increase the frequency of stimulation, if I overcome that refractory period, I can now lift it. Okay? And here I decrease, and here I increase again, and decrease, and I keep it steady, and I increase, and I decrease frequency. Okay, so during the relative refractory period, there is stimulus that can produce action potential. It just has to be stronger than the original one. Do you see what I'm saying? Does that make sense? Now, in the very beginning, when we were talking about the structure of the cells of the central nervous system, myelination and things like that, we mentioned that Schwann cells and oligodendrocytes provide electrical insulation to the cells. And I mentioned on past that it increases the transmission of the stimulus, increases the rate of transmission of the signal through the axon. Does that make sense? Signal goes faster. So now we're ready to figure out, the explanation. So generally speaking, if you do the local depolarization, if there are no voltage-gated channels located very close to each other, then this change in membrane potential will just decay. Like ripple will decay with time. But if the stimulus is strong enough and the voltage-gated channels are located close to each other, then depolarization of the membrane here, will cause the depolarization, that wave of depolarization will reach the next voltage-gated sodium channel, which will open and maintain the wave of depolarization. And this wave is keep going down the axon, okay? Sort of a domino effect. Depolarization that is produced by sodium channels at one patch of axonal membrane will open sodium channels at the next patch that will cause depolarization that will open sodium channels at the next patch. Essentially, it's the positive feedback. Okay, it's a classic positive feedback. However, you can imagine that the density of voltage-gated sodium channels on the axonal membrane must be pretty high. Does that make sense? And when every channel has to open, the transmission rate for the signal is going to be very slow. You see why? Because you have to open so many channels. Now what happens in myelinated axons, you have this electrical insulation right here. Myelin sheath prevents the dissipation of electrical charge from the membrane. Does that make sense? So the reach of this depolarization becomes longer, farther. So now you can separate ion channels by a larger distance. You can place them less frequently. You can place them essentially only in the regions of the membrane where myelin sheath is not present. You can put them in the nodes of Ranvier. Does that make sense? Essentially, what happens in myelinated neurons is called saltatory conduction. In saltatory conduction, saltatory means jumping. In saltatory conduction, the membrane potential jumps from one node of Ranvier to another. Does that make sense to you? So you have to know what the saltatory conduction is and how it works. Okay? Why it's saltatory? What is in the nodes of Ranvier? Why saltatory conduction doesn't happen in the non-myelinated neurons? Okay? What's going on in non-myelinated neurons compared to myelinated ones? Go ahead. In non-myelinated? On the very top or in the middle? Very top, you have no voltage gated channel, so it cannot spread. In the middle? In the middle, the problem is you have leakage of charges away from the membrane. When you... No, gates are actually maintained. Think about the electrical wire. Any electrical wire, any, should be electrically insulated. Did you see the cables? They're insulated. Why? To avoid you being killed? Oh, come on. Give me a break. Nobody cares. Okay? To avoid the loss of electricity from the wire. Does that make sense? So, if you insulate it, it's perfect. Okay? Electricity, you will maintain the same level of electrical flow through the wire for a longer distance. This is exactly what happens here. You electrically insulate the axon and, well, instead of maintaining the flow, you make it faster because you have to open less channels to maintain this wave of depolarization. And it plays an important role in understanding which type of signals will be transmitted by certain types of fibers. We had a conversation before that not every axon is myelinated, honestly. Some of them are, some of them aren't. Now think about this. Your muscle activity, your ability to contract muscles, is it fast? Can you contract them fast? Sure, yeah. Your response times are incredibly small, really. Does that make sense? So what does that mean in terms of the speed, rate of the signal as it goes from here to, say, here? Is that fast? If it's fast, what type of signal that is? Myelinated. That is correct. Does that make sense? Now think about your sensations. If somebody will stab you with a knife, are you going to feel the pain right away? Well, I'm not going to try, but imagine. If you step on the needle, okay? You can feel the prick, right? And then maybe later there's a dull pain. Okay? This dull pain happens seconds, sometimes minutes after the initial injury. The acute pain, the pain that you feel right away, is transmitted through myelinated fibers. The dull pain, that sensation goes through non-myelinated. It is a slow response. You see the difference? So you can actually kind of guess which ones to what. Right? Now, postsynaptic potentials. That's an official term for what's going on the neuron that happens, that is after the synapse. So think about this. You have synapse right here and synaptic cleft. And you can see in the synaptic cleft those really tiny dots. Those really tiny dots are neurotransmitters. neurotransmitters can be excitatory or inhibitory. When we talk about excitation, what does that mean in relation to the changes in membrane potential on the postsynaptic neuron? Excitation means what? Membrane potential on postsynaptic neuron becomes more, not stronger, more, increases, yes, becomes more positive. In absolute terms, in numerical terms, it becomes more positive. Does that make sense? Okay? Now, inhibition means more negative. And it makes perfect sense. Look, when the neuron is excited, when the membrane potential of the neuron becomes more positive, the neuron becomes closer to threshold. It's more likely to develop action potential. Does that make sense? When it becomes more negative, it is farther from the threshold, so it's less likely to develop the action potential. Does that make sense to you? So now, what can produce really? What really can produce normally the excitatory or inhibitory effect? Which ions? So what will lead to depolarization of the membrane? It's going to be only sodium. Based on the number of channels, it's the sodium influx. That's it. Does that make sense? So excitatory neurotransmitters like glutamate or dopamine or ACH in some cases, okay, acetylcholine, they will open the ligand-gated sodium channels. Does that make sense? You see what I'm saying? Neurotransmitters that are excitatory will interact with their receptors and open ligand-gated sodium channels. When ligand-gated sodium channels are open, where does the sodium go? There's only in or out of the cell. In the cell. Okay? Membrane potential becomes more positive, or less negative, which brings cell closer to the threshold. Can it cross the threshold? Potentially. Yeah, it can. Depends on how much neurotransmitter is released. Depends on how many receptors are there. Okay? It can. This potential, this little change, is called, this one that is shown in the picture, is called excitatory postsynaptic potential. In general, if you refer to this one, you know, the one that you can see on the picture, what you see is that a graded potential or action potential. Graded potential. On this exact graph, the change in the membrane potential did not reach the threshold. Does that make sense? Good. Now, what can bring the cell farther from the threshold level? What can hyperpolarize it? What can produce inhibitory postsynaptic potential? I have two options. One, bring negative ions in. That's going to be chloride. Does that make sense? Another example is to let positive ions out. That's going to be potassium. Okay, we good? So, obviously, you know, either way will work. Okay? And what I wanted to say, totally forgot. Anyway, so that's inhibitory postsynaptic potential. It's also graded, right? It's a graded potential. Okay? We good so far? Now, to kind of this part of our discussion is very physiologically dense. It's very fundamental. You know, we had some clinical connections when we talked about neurotransmitters, but now there are none. Now, I'm going to ask you a little question. So if I bring the neuron right here farther from the threshold, is that going to increase the chances to transmit the signal or decrease? So, decrease, right? The chances that the signal will go through the neuron are lower now. Does it make sense? I'm going to bring you the example from one of my favorite animals. It's the parasite, the worm parasite. Have you heard about the drug ivermectin? Have you heard about heartworm? Do you treat your pets for, well, dogs mostly, yeah, for heartworm? Well, you give your dogs ivermectin. And that's a fantastic drug. By the way, the guy who developed it got the Nobel Prize in 2016 or 16, I think, in physiology and medicine. They got Nobel Prize. William Campbell. So what this drug does is just, in my opinion, just absolutely amazing. The worm, it's not Toxocara canis. Dirophilaria, and I don't remember the species name. It's Dirophilaria gene. Anyway, it's a round worm that gets into the muscle tissue of the dog and destroys it and causes heart failure. Okay? And all roundworms have pretty well developed nervous system, which allows them to move. And in that nervous system, on the neurons, they have glutamate receptors. Okay, molecules that interact with a neurotransmitter called glutamate. We talked about it. And glutamate in worms is inhibitory. It opens chloride channels. Does that make sense? And that hyperpolarizes, opening chloride channels, hyperpolarizes neurons. When you treat your dog with ivermectin, ivermectin binds to those channels, to those receptors of glutamate. And what it causes is massive opening of these chloride channels. Does that make sense? So instead of a regulated transmission, you have a massive influx of chloride into the neurons of the neuron system of the nervous system of the worm. All neurons are what if chloride flows in? They become farther from threshold. What happens to the signal transmission? It's gone. The worm is paralyzed. It's not dead. It's paralyzed. That's why. Do you treat your dog once or once a month? So when your dog takes this treat once a month, it keeps the worm paralyzed. So they cannot reproduce, they cannot move anywhere. If your dog is infected, the worm is not going anywhere, but it cannot do much because it's totally paralyzed. Yeah, say again? Pretty much, yeah. Yeah, it'll die. It's going to take a while, but it's going to die eventually, yeah. Yes. And dog can inoculate, dogs are not cleanest animals. They coprophagic, and so they can inoculate themselves, but just keep giving, yes.
Can they ever get rid of it? Like,
The worm?
Once the worm is dead, yeah.
Oh yeah, it will eventually be destroyed, but look, again, dogs are coprophagic. They're gonna get new ones. That's why I just keep going with it. Now, as we just discussed, postsynaptic potentials, this thing, they are not necessarily action potentials. You see what I'm saying? At each synapse, and this is something I probably should have mentioned, and I'm going to mention it now. What you look at here and here, you look at the individual synapse. How many synapses are there on one neuron? I'm going to tell you, hundreds. Does that make sense? So you have many graded potentials at different places of the neuron at different times. So the question is, can they add up? And the answer is, of course. This process of adding up is called summation. Okay? Summation can be temporal or spatial. So temporal or temporal, I don't know what's the proper emphasis. Temporal summation happens when you have two stimuli, okay, two excitatory postsynaptic potentials happening so close to each other, you know, time-wise, that they overlap. Does that make sense? And I will give you some analogy. Spatial summation happens when you have excitatory postsynaptic potentials happening at the same time at the different locations on the neuron. Now, here's my analogy for this. Imagine that your car is stuck in the snow. Okay? So if you say this is the car, and this is what I'm going to do to get it out of the snow. I'm pushing it and I'm, moving back. And I'm pushing it and I'm going back. It's probably not going anywhere because it's stuck. And single effort like this isn't going to do much difference. Does that make sense? This is what happens when excitatory postsynaptic potentials arrive at the neuron one after another at the lower frequency. Now imagine I come to the car and start to push it very frequently. Does that make sense? It moves forward a little bit and I keep pushing, and it moves forward more and I keep pushing. So I increase the frequency of the stimulation. Does that make sense? I might eventually just push it out of the snow. That's the temporal summation. When one action potential overlaps another, maybe a third one overlaps it. When they start to overlap, the level of membrane depolarization exceeds the threshold, and you've got your action potential, you've got the signal. Does that make sense? Now, spatial summation is when you call your friends and you have like 10 people pushing the car out of the snow. They all do it at the same time at the different places, but at the same time. Does that make sense? That's spatial summation. Now you can imagine that each of the graded potentials, each of excitatory postsynaptic potentials, bring the neuron to the closer to the threshold. But only when several of them are combined, neuron produces the signal. Here's the example from, again, human physiology. We're going to talk about it again. Your eye. Can you see each other now? Can you see in color? Is the image sharp? Well, some of us wear glasses, but the image is generally sharp, right? Imagine for a second that we turn off the light completely in this room. We like block every source of light, anything. what's going to happen to the image? Even when your eyes adjust, will you be able to clearly distinguish colors? Probably not. What about the contours and sharpness of the image? Yeah, it's not going to be that good. Of the same person, at the same distance, no, it's not going to be that good. So here's the interesting difference between your vision in the light and vision in the dark. Cells that are responsible for your vision when the light is present, each cell generates its own sequence of action potentials. Does that make sense? That is why the sharpness is incredible. Okay? So your brain receives the information from each and every single cone in your eye. Each cone produces the action potential. Now, when light is off, cones have very low sensitivity. You know what I'm saying? So they require very powerful stimulus. You really need a lot of light to produce the image. Does that make sense? So when there's no light, cones are useless. Rods, on the other hand, they're very sensitive to the light. So they don't need a lot of light to produce EPSP. Okay? But here's the thing. About 200 rods together send the signal to one downstream bipolar ganglion cell. So what you end up with is 200 excitatory postsynaptic potentials from 200 rods combine on one neuron, and that signal goes into your brain. So when you have so many signals combined, sharpness of the image decreases. The resolution decreases. Does that make sense? But you can see in the low light conditions. Okay? So it's kind of, it's pretty much like, yeah, the more pixels you have per square centimeter, the sharper you are, yeah. So in this case, 200 rods, they represent 1 pixel. One cone also represents 1 pixel. So definitely if you have a lot of cones in a small space, you have a lot of pixels in a very small space. Okay. So summation often happens, but it doesn't have to happen. Another thing that can happen on the postsynaptic neuron is the situation when you have inhibitory and excitatory postsynaptic potentials coming together. In this case, they may just cancel each other out. Does that make sense? Now, the last thing before we going to break is neuronal pools and neuronal circuits. So far, when we were talking about the transmission of a signal between neuron and presynaptic and postsynaptic neuron, we presumed that they are interacting, one with another, and that's it. So I have 2 cells, right? In fact, neurons are organized in pools. So for instance, a given presynaptic neuron can extend its fibers to multiple neurons downstream. That makes sense? So when action potential travels down the presynaptic neuron, the amount of neurotransmitter that will be released from each branch, sorry about that, will be slightly different. Okay? In certain downstream postsynaptic neurons, the amount of the amount of neurotransmitter will be sufficient to produce action potential. This is called discharge zone. Okay? In others that are located in the facilitated zone, action potential will not be produced. What's going to happen instead is that excitatory postsynaptic potentials that will be produced in the neurons in facilitated zone will bring them closer to the threshold. And if they are closer to the threshold, summation can occur. Does that make sense? That's why it is called facilitated. The signal from that presynaptic fiber, right here, causes action potential in the discharge zone, so clear signal transmission, and also facilitates, helps, increases the chances for signal transmission in the neurons of facilitated zone. That makes sense? Okay. Right? Like if I would have a hose, start spraying you with water, people who will be right in front of me will get soaked the most, but people on the sides will get some splashes as well. They wouldn't be soaked as much, but so that would be my discharge zone, that would be facilitated zone. Now neurons are also organized in circuits, and different circuits of different structures generally regulate different activities. Diverging circuits, exemplified here, have one input which branches out to multiple outputs. You can see it here. It all starts with the neuron here, which discharges to this two. Each of them discharges to two neurons more, and they just keep branching down. Usually diverging circuits are responsible for transmitting motor signals, okay, like skeletal muscle. Does that make sense? So you can have one upper motor neuron, which will discharge to multiple lower motor neurons, which will stimulate hundreds of thousands of muscle fibers. Does that make sense? Converging circuits, on the other hand, can be found in the sensory transmission. When information is collected from multiple sensory inputs, multiple sensor receptors, and accumulated on one neuron, say multiple receptors like Merkel cells, transmit the signal to first order neuron, which then communicates it to the brain. But it's going to be multiple cells, so that allows to amplify the sensory signal. Or the example that I gave you, multiple rods transmit the signal to a single ganglion cell down the road, which allows you to see in the dark. Reverberating circuits, when neurons can signal back, okay, make it sort of a cycle, okay. In reverberating circuits, you're going to find them controlling repetitive activities. It can be motor activities like walking, running, swimming, or breathing, which is also classical repetitive activity. Does that make sense to you? And that makes, reverberating, going back in the circle, okay, repeating itself. Finally, parallel. Yes. it would be lovely to understand how sleep-wake cycle works. We have a bunch of proteins, protein signals that regulate sleep-wake cycle, hormonal signals that regulate sleep-wake cycle, serotonin regulates sleep-wake cycle, melatonin regulates sleep-wake cycle, potentially dopamine, as far as I remember, cholecystokinin can regulate. So histamine regulates sleep-wake cycle. So we have like a bunch of chemicals in the brain that participate in a sleep-wake cycle. And in addition to that, the light perception regulates sleep-wake cycle, which is reflected on melatonin production. So it's not just one circuit. It's multiple ones. And finally, parallel after discharge. These circuits can be found in the parts of the brain that are responsible for higher level processing. It's pretty much thinking. Information analysis, okay, like math stuff, calculations. Now, it kind of makes sense, because if you have multiple neuronal pathways working in parallel, it increases the processivity, it increases the speed at which you can analyze the information. Does that make sense? Understand the difference between discharge and facilitated zones? What happens in each? And please know which circuit is involved in which type of activity. I'm not going to ask beyond what I told you here and what is very shortly written here. Did I make myself clear? Very good.